Welcome, dear uveitis friends. Welcome to the 16th webinar, which is a very special one. It's a very theoretical one, but I think it's very important to have some ideas what's behind some of these diseases. As you know, you will get an evaluation sheet after the meeting, after this webinar. And if you send that back in between 1 week, you get CME points. Please use for your questions and answers only this part here, question and answers. Don't use chat. Chat is actually only for the faculty, for the speakers. And then I think I can give to Vishali.
Thank you. Thank you, Manfred, and welcome you all once again to our monthly webinars in uveitis. And as Manfred says, it is the 16th one, and we are now moving on to more in-depth and more complex topics that will help us understand why uveitis occur when it occurs, because ultimately these are the kind of the things and this kind of knowledge will help us giving a direction towards more personalized treatment, which many of our patients deserve. Welcome you all once again, and we have the best of the best speakers with us in the field. And without further ado, I would invite none other than Rachel Caspi to begin. She has two talks back to back. The first one would be animal models in uveitis, and second is microbiome and uveitis in experimental uveitis. So, Rachel, I'm not going to interrupt you in between. Please continue your one talk after the other. And request everyone to please type your questions in Q&A box that our speakers would try to answer after their talk. Welcome, Rachel, and over to you.
Thank you very much, Vishali. And as you said, I will be giving the first two talks, and I'm very pleased to have this opportunity to speak and participate in this webinar. I will talk about mouse models, and Dr. Gerhild Wildner after me will talk about models in rats. So I will first introduce the different types of autoimmune uveitis models in mice, the induced and the spontaneous models, and I will briefly talk about their characteristics and the inflammatory cytokines driving them, which we hope are representative of human uveitis, and their clinical implications. Now, my second talk will be on the role of microbiome, and I will propose that uveitis may be triggered by gut-resident microbes. I will show that human gut flora also can support development of uveitis in these mice. I will also show evidence that uveitis can be regulated by microbiota even after its induction, which suggests that there may be a clinical potential of targeting the gut microbiome. So on to animal models of uveitis. And I certainly don't need to introduce non-infectious or presumed autoimmune uveitis to this audience, but I wanted to mention that recently the autoimmune nature of non-infectious uveitis has been questioned. Namely, it has been proposed that the disease is not autoimmune, but rather is autoinflammatory. So arguments for autoinflammatory etiology are the genetic associations that have been recorded with allelic variants of innate autoimmunity receptors. For example, Behçet's disease is associated with NLRP3 or cryopyrin mutations.
Rachel, sorry, do you have sharing slides?
Sorry?
Slides. We are not seeing any slides.
We are not seeing any slides. That is very weird.
Yeah, because I thought you were introducing your talk. Can you go to your screen first?
So I am seeing my slides, and we saw my slides when we were trying before. So I have no idea what is going wrong.
So you have to sharing your screen.
I am sharing the screen, yes. Let me start again.
Yeah, please do that.
Share screen.
And then you click on the presentation.
Yes, yes.
Yes, here we go.
We
That's
are
it.
seeing your desktop, maybe not slides.
Okay.
Yeah.
Is
Perfect.
it visible now?
Perfect. Go on.
I
Perfect.
must not have shared the screen successfully, but it is
No
okay.
problem.
So my first talk will be animal models of uveitis. And my second talk will be the microbiome. And on to the first talk, animal models. And here we go. So as I said, we are all experts of uveitis here, so I will not introduce the disease, which accounts for about 10 to 15% of severe visual handicap. But I do want to introduce the controversy. I guess we're starting here with a controversy instead of ending, that the etiology of uveitis is not really autoimmune, as we had been thinking all along, but rather autoinflammatory. And that is based on genetic association of allelic variants of innate immunity receptors, such as, for example, in Behçet's disease, there are associations with NLRP3 mutations. And there is central involvement of IL-1 in pathogenesis of some uveitic entities. The arguments for autoimmune etiology are lack of an intraocular infection, strong genetic associations with HLA antigens, and most importantly, perhaps presence of immunological memory responses to retinal proteins, as well as central involvement of T cells in pathogenesis. But I would put to you that irrespective of the initial trigger, everybody is right. Autoimmune responses to retinal antigens that are released as a result of tissue breakdown may down the line become drivers of disease. For example, responses to retinal antigens have been reported in the prototypic autoinflammatory Behçet's uveitis. So we all know that clinically and immunologically, autoimmune uveitis is a heterogeneous disease. Both Th1 and Th17 cells were reported in association with different types of uveitis patients, potentially underlying some of the heterogeneity. But due to practical and ethical limitation, causal relationships, well, mechanistic studies overall are very hard to perform in humans. So we depend on animal models to perform hypothesis-driven studies. And unfortunately, no single animal model can reproduce the entire spectrum of human uveitis. So to study this complexity, we and others have developed animal models of autoimmune uveitis, which differ in clinical appearance and immunological characteristics, and may represent different aspects of human disease. And here are some examples of selected induced and spontaneous uveitis models in mice that are in use in our lab. So models of uveitis in mice, and this obviously is a partial list. We recognize two types of models, autoimmune and autoinflammatory. The autoinflammatory models are purely innate, like LPS uveitis, for example, which most of us know. And unfortunately, I will not have time to talk about them. They can be useful, especially for rapid screening of anti-inflammatory medications, but they cannot model chronic autoinflammatory processes. So I will concentrate on autoimmune uveitis, which is driven by adaptive T cell immunity. And among the autoimmune models, there are induced models, where we provide the antigenic stimulus, and spontaneous models. And each has its own advantages and disadvantages. I will only mention today the three most often used and arguably the most useful models. The classical uveitis model, where mice are immunized with a retinal antigen in complete Freund's adjuvant. Adoptive transfer of in vitro cultured effector T cells that have been isolated from these mice. And I will also talk about spontaneous uveitis in IRBP T cell receptor transgenic mice. IRBP stands for interferon receptor retinoid binding protein, and that is one of the prototypic retinal antigens that induce uveitis in animal models. So the classical uveitis model, the classical model of immunization with retinal antigen in complete Freund's adjuvant. Now, why complete Freund's adjuvant? Because T cells, which are participating in these responses, are fully activated for pathogenicity only if they are exposed to both adaptive, antigen-specific, and innate bacterial stimuli, the so-called danger signals or adjuvant effects. Now, the classical uveitis model is induced by immunization with the retinal antigens to which patients have recall responses, such as IRBP that I just mentioned, and S antigen, otherwise known as retinal arrestin, incomplete Freund's adjuvant, or infusion of T cells from donors immunized with these antigens. The pathogenesis involves Th1 and Th17 cells, which are induced at the site of immunization, and they are distinguished by their signature cytokines, interferon gamma and IL-17. Now, these cells migrate from the site of immunization to the eye. They break through the blood-retinal barrier, recruit inflammatory leukocytes, and induce what we know as EAU or uveitis. Quantitation of disease is on a scale of 0 to 4 by number and type of lesions. And susceptibility is strain dependent. Our most susceptible strain is the beta-NR3 strain depicted here. C57 is decent, and BALB-C is actually resistant. So the induced EAU model, which is probably the best known uveitis model in animals, serves as a platform for testing new therapeutic approaches. And Bob Nussenblatt, whom many of us know and remember, used to say that the ability of an immunomodulatory therapy to affect development and or progression of the experimental disease has often predicted its clinical efficacy. So here is a list of the therapeutic agents that had initially been tested on animal models, and they were all successfully translated to either clinical trials or actual treatments, with the possible exception of the anti-IL-17 therapy, but that is a story in itself, and it's a story for another time. So a variant of the induced model is the humanized model of uveitis, which is induced by immunization with retinal arrestin of HLA transgenic, class 2 transgenic mice, which have been knocked out from murine class 2. And retinal arrestin or S antigen elicits memory responses in uveitis patients. It's probably the best known retinal antigen that induces responses in patients, in patient leukocytes, but it's characteristically poorly uveologenic in wild-type mice. However, DR3 transgenic mice, DR4 and DQ8 transgenic mice develop severe uveitis with this antigen. And so this is an important proof of concept which supports an etiological role for retinal antigens in the human disease and which validates the use of the EAU model for the study of human uveitis. So, however, the induced uveitis model has limitations. It requires strong bacterial adjuvants, such as complete Freund's, which contains mycobacteria, or pertussis toxin, which are not physiological, perhaps, and may introduce bias. And this model is not appropriate to study natural triggers of disease because we provide the trigger, we immunize. So we have developed spontaneous uveitis, a spontaneous uveitis model in IRBP T-cell receptor transgenic mice. We call them R161H mice for historical reasons. So these are beta-NR3 mice that are genetically engineered to express a T-cell receptor that is specific to the epitope of IRBP 161-180, which is derived from a mouse immunized for uveitis in the current classical model. So we cloned the T-cell receptor from the classical model immunized mice, and we made these transgenic mice. And in these transgenic mice, 25 to 30% of their peripheral T cells are all specific to this epitope. Now, this is a very robust and reproducible model of uveitis with about with really 100% penetrance. Disease starts around between 3 to 4 weeks, which is around weaning age, and by 2 months, all mice have it. And eye-infiltrating T cells produce both interferon gamma and IL-17, so they are Th1 and Th17 T cells as we know them. So the question arises, which model, the induced or the spontaneous, is a better representation of human uveitis? So each has its advantages and disadvantages, obviously. The induced model is a very controlled model. Immunization provides a trigger, a simultaneous trigger that allows disease to develop in a synchronized fashion that makes it easy to study. However, the hyperstimulation of the immune system by the bacterial adjuvants may not be physiological and may skew the response. For example, cytokine preference of particular models may be influenced by the adjuvants that are used. The spontaneous model of uveitis, there is no need for immunization and no need for adjuvants, but mice have a higher than normal frequency of retina-specific lymphocytes, which is also not physiological. So we must remember that all models are by necessity amplified to provide a high penetrance of disease, or they would not be useful. If our mouse models had the frequency of human uveitis, we would need an animal house the size of Manhattan. So they have to be amplified, and that is something that is both a strength but also weakness of these models. So in the case of the induced and the uveitis models, we believe that both models may represent different aspects of human disease and may perhaps be used to address specific questions. So let us talk a little bit about cytokine dependence of uveitis. In the induced, as well as in the spontaneous model, both Th1 and Th17 cells are induced and both infiltrate the eye. We can see that here. So which effector response is more relevant to pathology of uveitis? So in the immunization-induced classical uveitis that I've presented, there is a strong requirement for IL-17 in pathogenesis, because if we neutralize IL-17, the disease goes away. But if we neutralize interferon gamma, it doesn't. Actually, it becomes a little more severe. And this pattern is also seen in mice that are knockout for interferon gamma or IL-17. So in this disease, in this model, although both Th1 and Th17 cells are present, as they are in other models, it is the Th17 response that appears to be driving the pathology. So onto the spontaneous uveitis model. And in this spontaneous model in R161H mice, full development disease appears to be dependent on interferon gamma. If we cross the R161H animals onto an IL-17 deficient background here, there is only a modest effect on the incidence and severity of disease. But if we cross them onto an interferon gamma knockout background, the disease is much more affected. Incidence is still 100%. It appears later, but the severity of disease goes way down. And finally, there is the adoptively transferred uveitis. So this is a disease that is induced in naive mouse recipients with activated IRBP-specific effector cells that had been isolated from mice that are immunized for uveitis in the classical model. And this disease, this model represents the effector phase or the efferent phase of disease after the pathogenic T cells had already been activated. So some people would argue that it has more clinical relevance than the induced models and the spontaneous models. So in this model, retina-specific T cells can be polarized in culture to Th1 or to Th17 by activation with antigen in the presence of a cytokine cocktail of the appropriate composition. And when transferred into naive mice, they both induce disease. They are both about equally pathogenic. So this tells us that both Th1 and Th17 T cells are each a standalone effector capable of independently driving disease. So to summarize what I have told you thus far, mouse models of uveitis differ not only in their disease manifestations, but also in their effector T cell dependence. The classical uveitis model is dependent on Th17. Spontaneous uveitis in the IRBP T cell receptor transgenic mouse shows a preference for Th1, and the model induced by adoptive transfer of T cells can work with either Th1 or Th17. So uveitis can be either Th1 driven or Th17 driven, depending on the model. And we believe that both Th1 and Th17 may be pertinent to human uveitis and perhaps may help explain some of the heterogeneity of the clinical disease. And in the literature, we have seen reports of both Th1 dependent uveitis and Th17 dependent human diseases. And in data that I have not shown you, we have demonstrated that effector dominance, Th1 or Th17, is actually determined by the inductive events or the environmental stimuli that are present at the time of the initial antigen exposure. So unfortunately, in human disease, we don't know what these early inductive events are because they happen before we ever see the patients in the clinic. So this, however, has implications for therapy, because if animal models are relevant to human disease, then targeting Th17 may be effective against some types of uveitis, whereas others may benefit from anti-Th1 therapy. But there's a problem, because although if one effector response may predominate, both are always present, and we know that these responses are plastic. They can morph into one another, sometimes at the level of the cell, but certainly at the level of the population. So suppressing one effector pathway could simply shift the response towards the other effector pathway, and we still get pathology, but now it's just affected by a different type of effector response. So the take-home message from this that I would like to leave you with is that targeting a single effector pathway in uveitis may not be sufficient. And we should consider therapeutic approaches that concurrently target both Th1 and Th17, such as ustekinumab or similar types of interventions that to offset this effector T cell plasticity. I will shift now to the microbiome and its effects in uveitis. So when you think of it, especially in humans, uveitis is actually a paradox, because we know from experimental manipulations that only activated T cells are able to enter the healthy eye by actively breaking through the intact blood-retinal barrier to cause uveitis. But retinal antigens are not present outside the eye. They are fairly sequestered in the eye because they are part of the visual cascade, and they have no business being outside the eye. So they are not present in the periphery to activate these circulating retina-specific T cells. So where and how are these circulating retina-specific T cells activated to be able to cause uveitis? So in the induced uveitis model, we know how they're activated. We provide an antigenic stimulus and complete for its adjuvant, which is heat-killed mycobacteria that provide the antigen-specific and the innate signals to activate these cells. But in the spontaneous model, what are these stimuli? So we reasoned that perhaps the gut bacteria, which are the largest source of microbes in the body, might have a role in providing these signals. And if this is indeed the case, do they provide only the adjuvant signals or also some kind of adaptive antigen-specific signals? So to make a very long story short, spontaneous uveitis in the R161H mice, and we have to use the spontaneous uveitis to answer these questions because that's where we can study inductive signals. So spontaneous uveitis in these R161H IRBP T cell receptor transgenic mice turns out to be dependent on commensal flora in their gut. And its elimination by oral antibiotics, starting from before birth, we put the dams on antibiotic treatment as soon as we know that they're pregnant, makes the uveitis not induced. And the same happens if we rear these mice in germ-free conditions where there are no microorganisms at all in these mice. So here is just the pathology in the different conditions. But if the mice that are germ-free are returned into normal, specific pathogen-free conditions and acquire normal mouse flora from their cage mates, they do develop uveitis. So there's nothing wrong with their immune system. And this shows us that commensal flora contributes to their disease. And the question is, how does it do that? What is the mechanism? So again, to make a very long story short, we see that activation of these R161H specific T cells occurs in the intestinal lamina propria by microbial products, and these cells that acquire Th1 and Th17 effector function. So what you see here is freshly explanted tissue, ileal tissue, from a 17-day-old pup that will not get uveitis at least for another week. But it already has cells in the lamina propria of the gut that show T cell receptor engagement by the NUR77 GFP T cell engagement reporter. Now, in experiments, in other experiments, we can show that R161H cells in the gut become Th1 and Th17 cells, and their presence is abolished by treatment with the antibiotic. So microbiota are required. And I'm not showing you these data, but endogenous IRBP antigen is actually not required. We just need the gut bacteria, because IRBP knockout mice also activate T cells in the gut to become Th1 and Th17 cells, even though they have no target antigen. And in experiments that I'm not showing you, we can show that gut contents extract, bacteria-rich gut contents extract, activates the naive R161H T cells in vitro and licenses them to induce uveitis when they are adoptively transferred to naive recipients. So for all intents and purposes, whatever activates these retina-specific T cells, and we don't know what bacterium actually provides that antigenic stimulus, it looks like a microbial mimic of the specific antigen IRBP that is present in the gut of these mice. And this shows that the retina-specific T cells in the intestine of R161H mice actually receive a signal through their specific T cell receptor that recognizes IRBP. And for these experiments, we crossed the R161H mice onto the TCR alpha deficient background. So in these mice, most T cells that are in their body carry the retina-specific T cell receptor, and they have no other T cell receptors that are not specific to IRBP, like the mice that I have been showing you until now. So these are purely T cell receptor, IRBP T cell receptor carrying mice. Now, they develop uveitis that is even more severe than that of the original R161H mice on the conventional background. That makes sense because they have more IRBP specific cells. They massively activate Th1 and Th17 T cells in the gut. And again, a broad spectrum antibiotic treatment attenuates the disease. And perhaps most importantly, for this proof of concept, immunohistochemical staining for phosphozap70, which is a molecule from the T cell receptor signaling cascade, this immunohistochemical staining shows that the retina-specific T cell receptor has been engaged. These yellow T cells are double positive for phosphozap70 and for CD4, demonstrating that they are actually actively signaling through their IRBP-specific T cell receptor complex. So this strongly supports a role for T cell receptor signals, but it certainly does not exclude a role for innate signals or what the so-called adjuvant effect, because microbes in the gut, being microbes, have a built-in adjuvant effect or innate microbial signals that they carry. So here's the million-dollar question. Can human gut microbiota support development of spontaneous uveitis in these mice? So for these experiments, we take germ-free parental mice maintained in individual germ-free isolators, and we reconstitute them with a fecal microbiome transplant by gavaging them with fecal material from 3 healthy donors. Let's just call them U, V, and W for short. So we now have 3 separate humanized breeding colonies that are established, and the offspring mice acquire human gut flora naturally from their parents. And then we collect eyes and tissues and fecal pellets from parental and offspring mice for analysis. So what do we see? So we see that colonized mice actually adopt only a subset of the original donor flora, but they preserve the microbial footprint of the donor. Here we can see by alpha diversity that each colony of the humanized mice retained fewer species than their corresponding human donor. And the beta diversity shows that in terms of composition, each colony remains closer to their original human donor than to any other donors. Most importantly, healthy human flora supports development of spontaneous disease in these R161H mice compared to SPF and compared to germ-free, which have little or no disease. But it happens to an apparently somewhat lesser extent than native specific pathogen-free flora. So what we observe is decreased frequencies of IL-17 producing T cells in these mice and somewhat increased frequencies of T regulatory cells in the intestines. And that was most prominent, the T regulatory cells in the U donor actually. And what is also interesting, that higher microbiome diversity in individual recipient mice appear to correlate with a higher disease score in these mice. Now, that is different than what has been reported for dysbiotic situations in disease states, where the microbiome actually was impoverished. But it makes sense, because if you have more different taxa, you have a better chance of finding that elusive antigen mimic that these mice, that these bacteria we think have. So we hope down the line to be able to examine patient flora in this model. We've only been able to examine so far normal flora, but normal people become patients, so they probably must have disease-inducing microbes before they become patients. And we hope to isolate microbes or microbe consortia that have maximal enhancing or inhibitory effects to understand what might enhance or inhibit disease, and to explore possible use of these inhibitory bacteria, perhaps as probiotics. So to summarize, I have shown you that commensal microorganisms provide a trigger and or an amplification signal to autoreactive T cells that drive spontaneous uveitis in this model. And signaling through the R161H T cell receptor specific to retinal antigen in the intestine appears to be involved, suggesting the existence of a microbial mimic. However, we do not discount that innate bacterial stimuli may participate and might also be important. I've shown you that human microbiota can support development of spontaneous uveitis, and further work, of course, is needed to identify the actual organisms and the actual antigens that are mimicking the retinal antigen, and to dissect the antigen from adjuvant effects in this model. But irrespective of these gaps in our knowledge, I think that implications for pathogenesis of human uveitis are obvious. And perhaps there are implications for pathogenesis also of other autoimmune diseases, because there's the huge diversity of microbiota in the gut. And if these bugs can mimic IRBP, they can probably mimic a lot of other tissue antigens. So we believe that activation of autoreactive T cells, T cell receptors by commensals may be a more common trigger of autoimmune disease than we appreciated until now. And since our initial data were published, many studies demonstrating mimic activity of gut microbiota were published in mice as well as in human diseases. And some of these identified the actual microbes and sometimes even the actual antigen, which we have not yet been successful in doing. So this provides strong support for the notion that microbial mimicry can indeed trigger autoimmune disease, even ocular autoimmune disease, glaucoma. So to finish, I would like to show some evidence, and mostly this is evidence from other labs, that in addition to acting as an antigen mimic to kickstart disease, microbiota can affect uveitis also through other pathways. So if we use the active immunization model now instead of the spontaneous, the disease trigger is provided. So any effects of the microbiota that we observe will reflect only antigen-independent effects. So in Phoebe Lin's lab at OHSU, Nakamura et al. Published in IOVS that EAU immunization of mice whose microbiota had been depleted by antibiotic treatment results in attenuated disease. Now, conversely, Pei-Zheng Yang and his collaborators showed that EAU immunization of mice that had undergone antibiotic treatment and were subsequently reconstituted by microbiota from uveitis patients results in enhanced disease. So they used microbiota from Behçet's disease and from VKH disease, and they followed the mice for 14 days. So this is the peak of disease. So this doesn't actually tell us whether this is a lasting effect, but certainly it does tell us that these uveitis patients had microbes that were enhancing for induction of uveitis. So these studies support the notion that manipulation of gut microbiota may be a viable approach in the future to therapy of uveitis. And I would like to finish by thanking all of our collaborators, friends, and colleagues who participated and influenced these studies. And I want to thank you for your attention.
Thank you. Thank you, Rachel, for very, very interesting talks. And I'm sure there's going to be a lot of discussion. And there are questions for you already, Rachel, in the Q&A. So we now move on to our next speaker, Dr. Soumya Basu, who will be talking about immunology of infectious uveitis.
So I am trying to unshare unsuccessfully.
That's okay. Just go on top of it, Rachel. There is a red button, a small thing which says stop sharing.
That is the button that I am looking for, and it actually has disappeared.
Don't worry, it will appear. Just run your cursor.
Okay, here goes.
Yes.
I think I have unshared my screen.
Okay.
Have I?
Yes, you have.
Thank you.
Audiovisual team, can we please start playing Dr. Basu's talk? Dr. Basu is currently enjoying Goa, so we have his recorded talk with us. We can start playing, please. Can you increase the volume? We can't hear.
When I started making this presentation, I realized that these immune mechanisms are really not so simple, because there are multiple infectious agents, which could be viruses, bacteria, or parasites. Each of these infections have multiple clinical manifestations which may overlap with other infections as well as with other non-infectious uveitis. And each of these infections can also have multiple pathomechanisms which may range from the replication of the microbes to the host immune response. So in this presentation, I'm going to provide an overview of the immune mechanisms in infectious uveitis, start with the microbial dissemination to the eye, then discuss the immune privileged nature of the eye, and follow this up with the outcomes of the host pathogen interactions. And then we discuss the mechanisms other than infection, which may have a role in the pathogenesis of infectious uveitis. And finally, the role of infections in non-infectious uveitis. So the microbial dissemination to the eye is best learned from the CNS literature, where we see that if the infection is outside the blood-retinal or the blood-brain barriers, then the blood vessels do not offer much resistance to these infections, and this leads to hematogenous dissemination in these tissues. On the other hand, if the infection is inside the blood-retinal or blood-brain barriers, then it needs several active mechanisms on part of the microbe to cross these barriers. This may involve infection of the endothelial cells, where these transcellular mechanisms allow movement of the organisms, or help of a phagocyte, which is used as a carrier to move across these barriers. There can also be several paracellular mechanisms where the junctions between the endothelial cells are disrupted to move across the barriers. Apart from these two mechanisms, there is also a mechanism of axonal transport, typically seen in the herpes viruses, where the viruses move through the neurons into the tissue involved. Now, once the organism reaches the eye, it meets a unique immune environment, which is called as the immune privilege. So how did this idea come into our minds? Now, the original concept was that anything foreign that enters the eye will be recognized by the immune system, and the immune system will react against it. But Peter Medawar performed this experiment in 48, and he showed that if skin allografts are put into the antechamber of the eye, then they do not meet with an immune response. Now, later on, it was realized that this phenomenon is not unique to the eye itself, but to many other tissues which have a low regenerative potential, or tissues where the stem cells are stored, for example, the bone marrow. Now, this immune privilege is not only the result of the physical barriers which we see here in the case of the outer blood retinal barrier, the retinal pigment epithelium, or the inner barrier which is formed by the endothelial cells and the pericytes, but also a very active mechanism which involves active immune suppression by release of several anti-inflammatory molecules which are listed here. Now, what is the impact of this immune privilege on the infection that reaches the eye? This is best exemplified in the story of the Ebola virus. You know, when the epidemic happened in West Africa in 2014, it was seen that patients who survived the viral infection were getting ocular inflammation. And this typically happened at least 2 months after the viral infection. Here, the viral RNA could be isolated not only from the aqueous of the survivors, but also another immune privileged site, which was from the semen. And this could be seen later on in the PREVAIL study in up to 40 months after the Ebola virus disease. So this shows how long active infection can persist in the immune privileged sites after the systemic infection has settled down. Now, the outcomes of these host pathogen interactions in the eye can be listed in terms of the organism we are talking about. And in case of bacteria, this is best exemplified by the Mycobacterium tuberculosis and its relationship with the retinal pigment epithelium. Now, it's very well demonstrated now that the RP cells are very good at phagocytosis of these mycobacteria, and they also control the MTB growth. The best thing is that, or the bad thing for the host is that the RP cells and the mycobacteria have a very nice relationship where they can survive with each other for a very long time. This was shown very well in the in vitro experiments performed in Professor Narsingh Rao's lab. So this forms the reason for the long latency of these bacteria within the eye. In case of viruses, it depends on what kind of virus we're dealing with. The DNA viruses, such as the herpes viruses or CMV, are very prone to latency, and the latency can happen in different cell types. For example, the herpes simplex or the varicella zoster virus has a tendency to be latent within the neurons, whereas the cytomegalovirus remains latent in the myeloid cells and the Epstein-Barr virus in the B cells. On the other hand, the RNA viruses such as Ebola and Zika, like we discussed in the previous slide, they have a tendency for persistence where they multiply in very small numbers within the organ, and this is typically seen in the survivors of the primary infection. The retroviruses also have a tendency for latency, and this is seen in HIV and HTLV, where HIV, for example, survives within the T cells of the host. For parasites, it's a very complex mechanism, and again, very well exemplified by Toxoplasma, where the T cells, the CD4, CD8 T cells, as well as the astrocytes, the oligodendrocytes, all of them work together to maintain the toxoplasma in the latent stage, which is known as the bradyzoid. And it's only when this mechanism is lost that the infection gets reactivated and we get the clinical manifestation. For fungi, Cryptococcus alone has a tendency for latency. The other ones, Candida, Aspergillus, these remain as commensals. And once the immune response goes down, they are prone to replication and tissue invasion. So the blood-retinal barriers actually have a major impact on what kind of etiology is causing the inflammation. For example, if you look at primary retinitis lesions, which are entirely inside the blood-retinal barriers, which could be due to viruses, parasites, or bacteria. Most of these retinitis lesions, apart from Behçet's disease, are caused by infections. And this is very well demonstrated in acute retinal necrosis or Toxoplasma retinochoroiditis. But what is not very well recognized is that it can also happen in tuberculosis. For example, this is a 19-year-old boy where we recognize this intraretinal granuloma so well delineated on the OCT scan here, where we could also identify acid-fast bacilli positive pulmonary tuberculosis in this patient, which was previously unrecognized. So even TB can have a primary retinal manifestation. On the other hand, the infection that is happening outside the blood-retinal barrier, that is typically due to lesser virulent organisms or lesser virulent genotypes. This genotype part actually we have studied in the zebrafish model of ocular tuberculosis. Cannot go into the details here. But in general, if you see that the infections are relatively uncommon when tissues outside the blood-retinal barrier, such as the choroid or the iris or ciliary body, are involved. And this is where the non-infectious conditions are way more common. Now, apart from infection, there are also other mechanisms that may have a role in the clinical manifestations. And this we realized when we looked at the literature on the histopathology of ocular tuberculosis, which showed that even eyes that require enucleation due to persistent inflammation, they also have a very paucibacillary nature where very few organisms are seen inside the eye. So we thought there must be another mechanism that is driving the inflammation in these eyes, and we looked at the T cells of these patients with tubercular uveitis collected by pars plana vitrectomy, and we realized that most of these cells are the CD4 positive T cells, and these are highly pro-inflammatory, and these have a cytokine response to both Mycobacterium tuberculosis as well as to retinal crude extract. Apart from this, we realized that the autoreactive T cells are also resistant to the activation induced cell death, which means that they persist longer inside the eye, and that is how they cause this inflammation. This is shown in this dot plot from the flow cytometry figures for those of you who can understand this concept. Now, when we think of autoimmunity in infection, the first thought that comes to our mind is molecular mimicry, but there can be other mechanisms as well, and these are that can be provoked by the microbial epitopes. And this is unrelated to the molecular mimicry, whereby one of the mechanisms is breakdown of the immune tolerance, which exposes the self-antigens to the immune system, leading to the inflammation. Now, it's not just that autoimmunity has a role in infectious uveitis. Infections also have a role in non-infectious uveitis. And to borrow from Charles Janeway's aphorism, this could be called as the ocular immunologist's dirty little secret. So the question is, how does a lymphocyte know when to proliferate and attack? Is meeting a cognate antigen enough to start proliferating? Here, it is infection which provides the context for the autoimmune response. So if there is no context to for the lymphocyte to act, it will not act just because it has met the cognate antigen. And this we heard very well in the previous two talks by Professor Ashley Caspi about the role of the Freund's adjuvant, which contains the killed mycobacteria in generation of experimental autoimmune uveitis, and also how we cannot have autoimmune uveitis in mice if they are germ-free. This is a model picture borrowed from actually a much later paper by Metzinger, where you see that the bacteria are reacting with the pattern recognition receptors on the APCs, and that is activating the T helper cells. So to conclude, there are multiple mechanisms that can allow dissemination of the microbes to the eye. This can have multiple outcomes because of the ocular immune privilege in the eye, which includes clearance of the organism, replication, and several situations in between, such as latency, reactivation, or persistence of the organism. Now, the blood-retinal barriers play a major role in shaping what kind of infections or non-infectious conditions are causing the inflammation. And we saw how retinitis is primarily caused by infections. Also, it's not just infections alone. Autoimmunity can also have a role in the inflammatory manifestations, and this could either be due to molecular mimicry or by bystander activation. Not much studies in the eye, but this would be a very interesting area for future research. And finally, infections too can have a role in the non-infectious uveitis by providing the context for the autoimmune response. Thank you for your attention and look forward to the questions.
Thank you very much, Soumya, for yet another very interesting talk full of all the learning outcomes for all of us model clinicians. And now we move on to another very interesting topic by Gerald Weltner, who will be talking about what can we learn from animal models for clinical uveitis.
Can you see my talk?
Yes, Gerald, all good.
Okay, thank you. So we now go back to animal models, and I have to thank Rachel Caspi for nicely paving the way for me. And I am only talking about our results we obtained from the rat uveitis from the last years. So experimental autoimmune uveitis can be induced in Lewis rats. It's an intraocular inflammation mediated by CD4 positive T cells. And those T cells per se are not pathogenic, but they have to recruit inflammatory cells. And in the rat model, they are mainly monocytes and macrophages. And those cells cause the inflammation and can destroy the retinal tissue. Rat EAU is a pan-uveitis, so we have anterior uveitis and posterior uveitis. And the disease can be induced in rats with several retinal autoantigens together with strong adjuvants. And our favorite autoantigens are S antigen and IRBP. And we use two highly pathogenic peptides called PDSEC from S antigen and R14 from IRBP. And of course, as Rachel told you, adoptive transfer of autoantigen-specific CD4 positive T helper cells also can induce uveitis in rats. We always had a problem. We know how to induce uveitis in animals, but how to induce uveitis in uveitis patients. As Rachel told you, naive T cells cannot enter the eye, and we do not have retinal autoantigens outside of the eye. We have a good tolerance. The blood-retina barrier prevents the immigration of non-activated leukocytes. In the thymus, ocular autoantigens are expressed, so we have a central immune tolerance, and the eye has several mechanisms for peripheral tolerance. So we postulated that we have to induce an extraocular cross-reactive immune response by antigenic mimicry of an environmental antigen.
Gerald, sorry,
yes?
Can I interrupt you? Your slides are not moving.
Oops, sorry.
Yeah.
This is strange. Oh, my screen sharing was interrupted. Okay.
Can you go back to screen sharing? Stop screen sharing and then come back to screen sharing. We see your first slide, but
I obviously was knocked out, partially knocked out of Zoom. This is crazy. Better? Do you see my slide?
Yes.
Okay. So I can go on. We are postulating that we are inducing uveitis in humans by antigenic mimicry, and we have some examples in rats. Here you see two snakes, a poisonous snake and a harmless snake, which have almost a similar pattern. If you know about the poisonous and the harmless snake and you see one of them, you have two types to decide. Either you run away because you are afraid, or you are very brave and you try to kill the snake, irrespective of whether it's the poisonous or the harmless one. And in case of autoimmunity, the immune system decides for the second way. It tries to kill. And indeed, we found some peptides which look very similar to the autoantigen peptide. You can see on the top row the peptide PDSEC from retinal S antigen. In the second row, a peptide from rotavirus, which normally induces gastroenteritis. And the third row is even worse. It's from bovine milk, a nutritional protein. And why can T cells get confused by these peptides? It's due to the T cell receptor, because the T cell receptor has a sloppy way to look at its antigen. It's only screening a few amino acid side chains of the antigen peptide and does not care what's in between. So with a few similarities, a T cell is content and will get activated. And so it can be activated against a huge variety of different peptides. And we could show in our rat model that immunization with the rotavirus peptide and the bovine milk casein peptide can induce uveitis in rats, and even the casein protein worked nicely. And it even works better when we did adoptive transfer of T cells specific for these antigens. But this was subcutaneous immunization, and this is not the way how you get in contact with these antigens. So we decided to try an oral immunization. This time we did not use CFA, but cholera toxin as oral antigen. And we were very disappointed because the peptides did not work. But to our surprise, the whole casein protein could induce uveitis in the majority of the rats. And here you can see the casein-induced uveitis is a quite severe disease. Then we looked at patients' immune responses to these peptides, and you can see here the antibody responses. Patients with anterior uveitis in black, with posterior uveitis in gray, and the white columns are the healthy donors. You can see the uveitis patients had enhanced antibody responses to PDSEC, to the casein peptide and the rota peptide. And you see the same with the T cell responses. So to summarize this part, peptide mimicry peptides rota and Cas are uveitogenic in rats. They are immunogenic in uveitis patients, so we get T cell and antibody responses. And those two mimotopes might be two examples of many, many mimotopes that are available that could induce uveitis or other autoimmune diseases. So foreign antigens can induce autoimmune uveitis, which fits also very nice to the microbiome hypothesis of Rachel, because the microbiome is living with us, but in reality, these are foreign antigens. So the next step was we looked for the migration of autoreactive T cells. For this purpose, we used GFP positive autoreactive T cells that we injected intravenously into naive rats. And then we watched the iris and the T cells appearing there. And we were surprised because 30 minutes after injection of the T cells into the tail vein, they had already shown up in the iris tissue. And they became more and more over the days, but we got nervous because we could not see any signs of uveitis. Then, after 3 days, we saw the inflammatory cells infiltrating, which are the clinical signs of uveitis in our model. And this phase, when the first T cells are already in the eye tissue until they are starting to recruit inflammatory cells, we call the prodromal stage. And this is what sometimes patients experience. And if those patients who have this certain organ feeling in their iris, if they start treating themselves with corticosteroid eye drops, they could prevent or at least weaken a relapse of their disease. Interestingly, was also in these experiments that only a very few autoreactive T cells here in bright green are necessary to induce uveitis, and former data by Rachel Caspi and Bob Prendergast have shown that you need only 9 autoreactive T cells to induce uveitis in a mouse eye. So this was only part of the story, because all activated T cells can enter the eye, but not all can induce uveitis. In another control group, we have injected activated ovalbumin-specific T cells. It's hen egg ovalbumin, which has nothing to do with the eye, and of course, it's not pathogenic. However, if you provide the antigen for those cells, which means you inject ovalbumin into one eye and inject the control protein like bovine serum albumin into the other eye, you can indeed induce uveitis, but only in the eye that was injected with ovalbumin. So as soon as the T cells see their respective antigen in the eye, they get reactivated. And you can see ovalbumin-induced uveitis is also quite severe disease. So the T cells must be reactivated by their respective antigen. A mimotope works as well. And what's also interesting, and which has been published by Igal Gary and his group many years ago, and we found the same, the T cells have to pass the blood barriers to become pathogenic. If you directly inject the autoreactive activated T cells into the eye, nothing happens. You will not get uveitis. So there must be a role of the autoantigen itself, because we found similarities and differences between the disease induced with the two peptides. PDSEC is a 14-mer peptide from retinal S antigen, which is an intracellular protein. R14 is a 21-mer peptide from IRBP, which is an extracellular antigen. And those peptides are presented to T helper cells by different MHC class II molecules in the rat. And we induce the disease in the same strain of rats, namely Lewis rats, so we do not have to care about any genetic differences. We induce the disease by similar immunization. We use the peptide antigens. We use CFA, the same site of injection. And we get the same clinical pictures. We get a pan-uveitis from hyperemia of iris vessels to hypopyon and hemorrhage. But there are differences between PDSEC and R14-induced uveitis, because when you immunize the rats or adoptively transfer PDSEC-specific T cells, you see one course of disease, and then the clinical signs are gone, and they are not reactivated. However, when you immunize the rats with the peptide R14 or do adoptive transfer, you see spontaneous relapses in both eyes. And then we looked at the T cells specific for the two antigen peptides, and we found that in T cell lines specific for R14, 29 genes are upregulated compared to PD-6-specific T cells. And those upregulated genes belong to different signaling pathways, but interestingly, all genes are up or downstream of interferon gamma. Then we looked at the autoreactive T cells in EAU and their cytokine production. This work was done by a former PhD student of mine. And she looked at interferon gamma and IL-17 producing cells in the eyes and lymph nodes of the PD-SEC-induced uveitis and the relapsing EAU induced with R14. And she found that only in the eyes, many T cells were producing interferon gamma and IL-17, and also both simultaneously interferon gamma and IL-17. In the lymph node, she only could see interferon gamma producing cells, very few IL-17 producers, and almost no double producers. So this is something very special for intraocular T cells during uveitis. And then she looked at the intraocular T cells at different stages of uveitis and their interferon gamma and IL-17 production. And in PD-SEC-induced disease, she found that interferon gamma IL-17 co-producing T cell populations increased from onset to resolution of disease. And this was amazing. And we thought maybe those double positive cells could be regulatory T cells because after resolution, there was no more relapse of the disease. It looked differently in R14-induced uveitis because we found an increase of interferon gamma producing cells and a decrease of IL-17 producing cells, but the population co-producing both cytokines remained quite stable. So we have a high number of IL-17 producing T cells in the beginning. So obviously these cells are necessary to bring in all the other T cells to the eye, but interferon gamma T cells play a major role later on. So Ulrike then investigated the role of interferon gamma in R14-induced uveitis. So she immunized the rats, waited until the first course of disease had resolved, and then she injected PBS or interferon gamma either intraperitoneally or into the eye. And she found that only in those groups where she had injected interferon gamma into the eyes, she sees a synchronized relapses. So interferon gamma is involved in relapses of uveitis and plays a pivotal role. So then she made experiments to investigate whether relapsing or monophasic or chronic uveitis which of them is dominant. And for this purpose, she immunized the rats either with a mixture of both antigens, or she separately immunized the antigens in contralateral legs. And it turned out that in the mix immunization, the relapses were completely gone. And in the animals that received both antigens in contralateral sites, the number of relapses was dramatically reduced. So the PD-SEC-specific monophasic disease is obviously dominant. And this could also be seen with the FOXP3 positive Treg cells, because the Treg cells were high at number in the mix immunization and in immunization with a separate application of both antigens. So there seems to be a bystander suppression induced by PD-SEC, which also works on R14-induced uveitis. I showed you that T cells are co-producing interferon gamma and IL-17 in the eye, but this was only part of the story because we also found a co-production of both interferon gamma, IL-17, and the suppressive cytokine IL-10 in eyes with EAU. And when we looked at our 14-induced uveitis, those triple-producing cells went down. At peak disease, they were very low. During resolution, they increased, and during relapses, they were low again. So these are probably regulatory T cells, because we saw the opposite in PD-SEC-induced disease. They increased during the course of the disease, and they were highest during the resolution. We've looked at further secreted cytokines and chemokines by those two T cell lines, and it turned out that the R14 specific T cell lines tend to produce more interferon gamma, while the PD-6 specific T cell lines produce more IL-17. And there was even more because R14 specific cells produced more IL-18, CCL3, and CCL5. While the PD-6 specific T cells tended to produce more IL-10, IL-6, and VEGF. And VEGF was very strange because our paper was the third publication dealing with VEGF secretion of T cells. This was very new for us. And the VEGF secretion of PD-6 specific T cell has consequences because about 4 weeks after immunization with PD-6 in CFA, or after transfer of PD-6 specific T cells, we see the growth of new vessels in the retina. So the question was, is this a reaction of the destroyed retina, or is it due to the autoreactive T cells? PD-6 specific T cells, R14 specific T cells can destroy the retina as well, but they do not induce neovascularization. So we set up another experiment. We have a small molecule inhibitor of activated T cells, and we tested this inhibitor on neovascularization in PD-6-induced uveitis. So here you see the vehicle control. And when we treated the T cell, or we inhibited the T cell at the start of uveitis, we dramatically reduced the number of new grown vessels. And even when we treated at the peak of uveitis, when it was too late to prevent any destruction of the retina, we still could see a significant reduction of growth of new vessels. So the neovascularization is clearly caused by the PD-6 specific T cells. It's not a tissue reaction to the destruction, because the R14 specific T cells cannot induce those neovascularizations. Last story, what happens to the autoreactive T cells in the eyes? This was also a surprise for us because about 5 to 6 weeks after injection of GFP positive PD-6 specific T cells, we still saw the autoreactive T cells in the retina, although we had no relapse. The same is for our R14 specific T cells without a relapse. But when we injected GFP positive R14 cells and we caught them at a relapse, we saw a clustering of the GFP positive T cells. Maybe this is clonal expansion, but we had no chance to investigate it so far. And as I told you, also T cells which are not pathogenic but activated are allowed to enter the eye. Here, GFP ova specific T cells. 3 weeks after adoptive transfer, of course, they cannot induce uveitis, but they are still in the retina. And this was 8 weeks after adoptive transfer of non-GFP R14 specific T cells. So you cannot see the autoreactive T cells. But then we injected GFP ova T cells. And 3 days later, we experienced the relapse. And we saw that the GFP positive T cells are somehow accumulating at the lesion of the retina. So the question is, are they preferentially accumulating at the site of reactivation or former scars, or did we induce the relapse by injecting the activated ova-specific T cells? So all activated T cells that have entered the retina remain there for many weeks, irrespective of active uveitis, and they can form clusters or accumulate at the sites of inflammation. To summarize, what can be learned from animal models for clinical uveitis? The induction of uveitis can be antigenic mimicry, so an environmental foreign antigen can induce immune responses that are cross-reactive with retinal autoantigens. The migration of autoreactive T cells into the eye is some days prior to clinical onset of uveitis, and this could explain the prodromy that some patients experience. We have similar clinical pictures, which is inflammation, despite the different immunopathogenesis, the relapses are caused by interferon gamma, so Th1 cells, and the IL-17 producing cells, which I'm not sure whether they are really T helper 17 cells. They might be regulatory in uveitis, which could explain the poor effect of anti-IL-17 therapies in uveitis. Because, as Rachel already mentioned, we have to deal with two types of T cells, which can change during the disease, and depending on which T cell type we are targeting, the other one can escape and keep the uveitis going. The dynamics of the intracellular T cell populations during uveitis are dynamic. We have a change of phenotypes and probably also of function. And this could explain the treatment failure if just one T cell type is addressed. Autoreactive T cells can secrete VEGF and can induce neovascularization in uveitis. So if we are effectively targeting the intraocular T cells, in that case, we can prevent the new vessel growth, and we do not need the VEGF blockers with their side effects. And the autoreactive T cells remain in the eye even after resolution of uveitis. They can form clusters or clones during relapses, and the previous sites of T cell infiltration are attracting new infiltrating T cells. So even when the patient is in a uveitis-free period, do not believe that the autoreactive T cells are gone. I want to thank my collaborators, last but not least, our rats, and I want to thank you for your attention.
Thank you very much, Carol, for very, very interesting coverage once again on the topic. There are quite a few questions for you in Q&A, which you can answer till the time we move on to our next speaker, who is Shona Cooper, who will be talking about immune networks of human non-infectious uveitis. Over to you. And your slides are visible, but can we please? Yeah, perfect.
I think you can hear me right now.
Absolutely, absolutely.
Perfect. So, so now we move on to discuss some of the immunological insights we have in human non-infectious uveitis, or in the actual patients. And so the great talks that preceded this one already introduced some of the key concepts of understanding pathology of uveitis in general. And I understand I made myself quite a challenge to explain to you some of the, let's say, relatively novel concepts of viewing the immune system as a network. So if there's one take from today that I want you to remember, is that the take-home message is that the immune system functions as a network, and that we start to believe more and more that that's also the case in non-infectious uveitis. So in contrary to the beautiful mechanistic work you can do in model systems of uveitis, as Rachel explained, it is usually quite difficult to do mechanistic studies in humans. So therefore, we have to apply a whole plethora of usually high throughput technologies to try to capture the signatures of the immune system and what is dysregulated in patients with non-infectious uveitis. Now, in Utrecht, what we usually try to do is combine patients' cohorts with well-defined types of human non-infectious uveitis, and apply a whole range of informatic layers and try to digest this usually with computational immunology. Now, this concept of the basis of T-cell immunity and also the involvement of antigen presentation by dendritic cells is basically still the classical view that is widely considered to drive also uveitis in patients, in humans. And this is based obviously on a large body of evidence from observational studies. But I think if we talk about some mechanistic proofs, is in particular that the whole network that is provided by cytokines that are linked to dendritic cells and T cells Some of them have quite some success in clinically alleviating the effects of inflammation in uveitis. Let me add the pointer to it. Yes. There, as mentioned, there has also been some drawbacks in the understanding of this. For example, the targeting of IL-17. That's obviously a different mechanism involved. But for example, one of the upcoming ones linked to IL-17 pathways might also be the inhibition of IL-23, one of those cytokines. Maybe more well known are the anti-TNF and anti-IL-6, which are also more widely used in refractory cases. So as I mentioned, the evidence for involvement of certain pathways in human non-infectious uveitis is usually circumstantial. So before I try to dive into some controversy as a way of looking at the immune system, let me definitely appreciate the fact that there is evidence to support the involvement of this axis. For example, numerous studies have looked at cytokine expression profiles in cases with uveitis. They intend to find many pro-inflammatory cytokines, including IL-17 and IL-23. There's infiltration of T cells in eye fluids, for example. Also in our lab, we've studied several types of those patients, and that usually includes also CD4 cells. Now, what you also see is that T cells, I'm not sure if this thing is in a way, but let me put that here. What we usually see is that the T helper 17 cells are increased in circulation of patients with uveitis. And we have some evidence that's also linked to let's say, proxy for disease severity for those cases that need treatment with immunosuppressive therapy. So I'm not sure if this thing is in the way here. Can you see my complete slide? Well, I just assume that's the case.
Yes, we can.
Perfect.
We can see.
And it's just me.
Yeah.
All right.
Good.
So also the basis for dendritic cells is circumstantial, obviously, and this is mostly work from other collaborators. This is a less explored field compared to T cells. But if you look at eye tissue of eye fluid, there's cytokines linked to dendritic cells and also infiltration of certain dendritic cell populations in the eye. And if you study, for example, blood composition, you indeed see also changes in the abundance and frequency of classical subsets of dendritic cells that are all considered to be key in driving T cell activation in human and non-infectious uveitis. So there's quite some evidence to support that also the observation that are made in the animal models resonates in what we see in patients. One other way of looking at this is, and this is obviously a very underrepresented slide of genetics in the field of non-infectious uveitis. If we summarize some of the things that seem to replicate throughout larger studies, is that we see evidence for antigen presentation, perhaps via dendritic cells, and the involvement of pathways and genes linked to T cell immunity. Now, one of those things is which you widely see in inflammatory conditions, and that's the enrichment for certain HLA alleles, so the presenting molecules on the cell surface, both class 1 and class 2, in all sorts of types of non-infectious uveitis, and the involvement of genes that actually process peptides, like Arabs, that's more linked to certain specific types of uveitis, but also the widespread linkage with cytokine receptors, and probably not coincidentally, cytokine receptors linked to functions that maintain T cell responses, for example. I must say, again, this is definitely an underrepresentation. There are way more genes linked to this, but this is also just adding to the body of evidence that this axis is probably also involved in human pathology of uveitis. So you could say, if we look at the immune system schematically, that this is definitely a section of interest for further prioritization of studies or drug targeting, as to say. The problem is that, well, it's not a problem, but if we dive into the complexity of the immune system in humans, we tend to see that there's actually starting more and more evidence coming out that there's more beneath the surface than just the interaction of T cells and dendritic cells. And there's many underexplored parts of the immune system And the number of papers that are slowly rolling out, showing that there's all sorts of other cell subtypes that have also some changes in abundance and perhaps linkage to uveitis in general. Now, another thing that is complicating clinical practice is the patient heterogeneity. Perhaps from clinical practice, for those clinicians among us, know that a typical uveitis patient can have quite different treatment responses or disease course, even when they have the particular same type of diagnosis, for example. And it's quite difficult to predict certain outcomes for that. Now, one of the holy grails, and this is definitely not restricted to uveitis research at all, or the domain of ophthalmology, is trying to better predict such things for individual patients. And one of the, somewhat, it's a word we nowadays don't like to use anymore, but for a long time we looked for biomarkers to better predict for individual patients and conduct personalized medicine. One of the major problems is that we can really state that throughout literature we have a tremendous biomarker crisis. So there's such a large amount of money invested in identification of clinically relevant biomarkers that then can be applied to stratify patients for treatments or whatnot. And the problem is that usually at the application part or the implementation part, and these biomarkers do not seem to perform as well as we hoped in the initial parts. And that actually let immunologists in general think about some of the concepts that are usually related to the design of biomarker studies, but perhaps also change a certain philosophical thinking of looking at the immune system. And this is, again, maybe one of the things I tried to propose to you for today, is that we try to think of the immune system really as a system. That's why it got its name. And this is a system that usually operates as a network. So although we definitely like to look at the immune system as having separate cells, separate functions, and that we like to study and that definitely are important, it's good to think about that the immune system has a lot of interacting compartments that probably all play a role, and might it be minor or major roles in conducting immune responses in non-infectious uveitis, for example. So basically, we have to move away a little bit from the scientific standard of thinking about pathways that are directional in such a sense. But if we want to truly capture the biological complexity, we need to think about networks. Now, and before you're starting to get headaches, this is typically a thing also for me in the beginning. I thought, whoa, it sounds complex, sounds something for computational people. How much does this apply to clinical practice? Well, I hope to give you an example today that at least makes you curious to think about the immune system as a network that we should consider for in our studies of non-infectious uveitis. So this also means that we have to be prepared to take a little bit distance from the textbook type of thinking, where we consider pathways as cascades that are directional or bidirectional, but start thinking about networks or maybe even circuits that could have intricate and unexpected interactions at all levels of the cascade as a whole. Now, to give you one example of where this evidence is based on, and this is actually from a recent paper using single-cell RNA sequencing of an aqueous humor droplet from several patients with HLA-B27 anterior uveitis, a common type of uveitis globally. And the point is, this is a technology that's able to capture the transcriptome of individual cells. This is a beautiful technology that can basically tell you unbiased, well, between brackets, unbiased, what do we actually see at the site of inflammation? And the take home from this paper, which I encourage you to look up, is that if you look at the inflammatory situation, we intend to see that the infiltrating cell seems to be a part containing the majority of immune cell types, obviously dendritic cells and T cells, but perhaps also many immune cell subsets which we not immediately consider to be involved in uveitis, or at least underexplored. Now, these are all technologies that are in the domain of omics, so high throughput technologies that basically gather big data, molecular big data, that help us to grasp this wide universe of information that is usually part of the network of the immune system. And these omics, which are definitely quite labor intensive, they need high expertise. They are expensive, but with the introduction in the last decades and the really accumulating and rapidly expanding number of omics that are out there, I believe that in the next coming decade or so, we will have this system-wide understanding of the immune system in individual patients. And we can harvest this information to better digest what is actually happening underneath the hood of patients that enter our clinics. But one thing to keep in mind here is that it's very important that we not gaze at one particular platform itself. And nowadays we see that major high impact papers seem to focus on single-cell RNA-seq, which is a beautiful technology, but keep in mind that the power of information is usually when it's complementary. So this is a schematic overview that the power of such network analysis is basically by using complementary parts of information to build a network like that, and hopefully see what is the immune system at work, where we can deviate and extract information from for further mechanistic analysis, for example. So why should we actually integrate multiple data layers? Well, this is a parable from the blind and the elephant. It's basically that if we only take one particular platform that we like and use, we may not grasp the entire picture or complexity of the problem at hand. Now, do we know if this network analysis actually work? Well, there's several beautiful studies in much larger populations that have conducted this previously, using large population studies of blood proteins and basically show that they can find robust and actionable targets for therapeutic targeting. Now, I will give you an example where we basically tried the same thing in a cohort of patients with uveitis. And we're not diving into the exact technology, but I will just dive into the main, let's say, conceptual overview of using network-based analysis in this. So this is a cohort of non-infectious uveitis patients. For those interested, here we have its anterior, intermediate, and posterior. And the whole idea is that we capture proteins in serum at diagnosis. And for the sake of doing a proof of concept study to find something clinically relevant, we were interested to see if we can use these networks to, let's say, stratify a risk for the use of immunosuppressive therapy, systemic therapy, later on in follow-up. Now, the whole idea here is that we used a relatively simple concept of network analysis. And that's based on the assumption that if proteins have more or less a same pattern of expression, they must have the same function. For example, in a pathway, or they represent something that's a cell type. And that's basically really based on the concept of correlation. So proteins that are relatively having the same pattern, we assume that they are also having the same function. And we can cluster that out of the network and use that then for downstream analysis to better understand what's going on. To give an example, and this is summarizing a lot of data, which I will send the link later on for you to dive further in. But we were able to find in this cohort, using about 1,000 proteins, a cluster of about 85 in this network. And the key take home here is that the level of expression of this cluster of proteins was relatively low in controls, but variable among cases with uveitis. So here you see the expression levels of 85 summarized, and you see it's actually variable among different cases with different diagnoses. Now, if you look at the follow-up data from these cases, which was on average about 2 to 3 years, it's quite interesting to see that higher levels at diagnosis seem to indicate that you have a relatively higher risk for starting immunosuppressive therapy during follow-up. We used all sorts of confounding analysis to see how robust this data was related to the serum signature. Now, the thing is, it's quite interesting for an immunologist to do all of these tricks with network analysis, but what can you do with this type of information in a clinical setting? So what we try to do next is use this network data and then start thinking about reducing that information to something that we can apply in simple terms, and that's available in clinics. We were lucky that this protein set was highly enriched for neutrophil proteins, We used all sorts of evaluation steps to assure that the proteins that we detected that were linked to immunosuppressive therapy were indeed highly expressed in neutrophils. And one of the convenient things was that we found also a reasonable correlation between the expression of the network and the neutrophil abundance at diagnosis. So this helped us to basically take a step back and use replication cohorts. This is a cohort from the Netherlands, where we basically used the baseline neutrophil count and evidence from the information we had from this network analysis and stratify cases and follow them up. And this basically showed that something as simple as your neutrophil blood count can help stratify the risk for immunosuppressive therapy later on. We also asked the team from Nidasen in the US to do precisely the same thing with the same thresholds, and they validated our results, showing that the power of network analysis to eventually also apply something clinically relevant for the patients with uveitis. And perhaps also unexpectedly involving, I was expecting T-cell cytokines, for example, but apparently neutrophils can also be relevant in certain tools for uveitis. So if you're interested, the preprint is out. You will probably can find it somewhere on MedArchive. As a last example, I also want to highlight that this is just an example of one omic technology, but I said, there was actually more, it's actually beneficial to integrate multiple of such information layers. And I fully agree. We basically also tried to do this, and this is the last example. It's quite a technical point, but the point is that if we use network-based analysis and we apply such network-based with certain types of omic data, we're also able to explore the immune system and perhaps even identify cell types which were not really well characterized previously. So we've put another preprint out where we used analysis in 2 cohorts of non-infectious uveitis patients and used the power of network-based analysis to eventually identify let's say uncharacterized dendritic cell population that in itself was had the characteristic of producing high levels of TNF-alpha and IL-6 that are known to play also a role in the pathogenesis of non-infectious uveitis in humans, as illustrated by therapies that suppress these cytokines. So the take home for today is start thinking about the immune system really as a network, and probably that you should also consider the fact that all these key compartments together collectively and dynamically move towards causing pathogenesis in patients, and that resolving this network will probably also be critical to further alleviate inflammation and start curing patients somewhere in the coming decades. Thank you.
Thank you very much, Jonas, for a very, very interesting concept. I think this is going to be future for all of us, and we look forward to more discussion on this. And now we move on to therapeutics. How do drugs influence the immune system in uveitis? And we have Hank Kaplan giving us the last talk of this session. Over to you, Hank.
Vishali, thank you very much. My talk is going to be on how drugs influence the immune system and cause uveitis during checkpoint block inhibition. There are 2 sections in the presentation. The first is cancer immunosurveillance and immunotherapy with checkpoint block inhibition. The second is ocular immune-related adverse events with immunotherapy. What is cancer immunosurveillance?
Hank, may I request you to please share your screen with us?
Oh my goodness. Okay. Hold on.
That's okay. That's fine.
Yes.
Yes. Hank, you can just click on from beginning. Yeah.
Yeah, okay. Okay, so I'll pick up where I was just a moment ago. The question is, what is cancer immunosurveillance? Cancer immunosurveillance is a natural function of the immune system and includes detection and elimination of transformed host cells. Thus, enhancing immune function can amplify anti-tumor responses and aid in the eradication of existing cancers and metastases. The subject of cancer immunoediting, by the way, there is something wrong with the presentation for a moment. For some reason, it's gotten.
Yes.
Unfortunately, it's not allowing me to share the.
Maybe you need to move the arrow on the left-hand side to move it forward. Sometimes it works that way.
I'm sorry, move what forward?
There is an arrow, a small arrow on the left corner of your screen. Yeah, that's right. It's showing.
Okay. The concept of cancer immunoediting was first introduced by Bob Schreiber in 2011. Because of time, I won't be able to go over the various steps in detail, but what I would like to show you is that, normal tissue can become transformed. The steps at that point, the host immune response can either eliminate the tumor or it can escape elimination by the host, or in fact, the tumor can exist in equilibrium. In point of fact, extrinsic tumor suppression is indeed what we would hope. What are immune checkpoint inhibitors? Immune checkpoint inhibitors are a class of bioengineered anti-cancer agents that inhibit the ability of tumor cells to suppress the immune system. They act by releasing inhibitory brakes on immune cells so that anti-tumor responses are promoted. However, the dynamic balance between self-tolerance and anti-tumor effects is very delicate, and disruption of the immunoregulatory roles of PD-1 and CTLA-4, two prevalent immune checkpoints, has the potential to cause autoimmunity. What are requirements for effective tumor immunity? Well, there are several. Tumors must express antigens recognizable by CD4 and CD8 positive T cells and or B cells. Lymphocytes must receive adequate co-stimulation from the tumor itself or from professional APC, antigen-presenting cells that present tumor antigens, or lymphocytes must avoid immunoregulation and suppression and have access to the tumor microenvironment to exert their effector function. What are the strategic choices for immunotherapy in cancer? There are two major choices, and that is to impact the innate immunity or adaptive immunity. And I will just focus on adaptive immunity and specifically active intervention through immune checkpoint therapy. There are several targets and drugs that are now available as checkpoint inhibitors, but the ones that are most common are the targets CTLA-4, PD1, and PD-L1. The table shows you the generic name of monoclonal antibodies to each of these targets, as well as their brand name. And there is a very nice review of this by Emmett Cunningham in Ocular Immunology, Ocular Inflammation and Immunology in 2020. CTLA-4-mediated inhibition of T cells can be prevented by a monoclonal antibody-based immunotherapy. As you see in activation, engagement of the T cell receptor is obviously important. In this schematic diagram, you see being presented by an antigen-presenting cell. So this represents the T cell. This is the antigen-presenting cell. In the process of activation, what occurs is that intracellular stores of CTLA-4 are expressed on the cell surface of the T cell. This then can compete with the co-expression of CD28 with B7-1 and B7-2. However, the use of anti-CTLA-4 monoclonal antibodies will in fact inhibit the effect of CTLA-4 and will result in enhanced T cell activity, which results in effective therapy. What was the first FDA-approved immune checkpoint inhibitor? It was ipilimumab, which is an anti-human CTLA-4, and it was designed for metastatic melanoma. In a phase 3 trial, it showed a survival benefit with ongoing responses for over 4 years. FDA approval occurred in 2011. Additional indications for ipilimumab now include not only melanoma, metastatic melanoma, but also ovarian, lung, and kidney cancer. In addition to a checkpoint inhibitor like CTLA-4, there's PD-1. PD-1-mediated inhibition of T cells can also be prevented by monoclonal antibody-based immunotherapy. And again, in this example, this represents the T cell, but this time instead of an APC, this is the tumor cell. And the tumor cells all have antigen-specific epitopes that can be recognized by the T cells. But PD-1 is also expressed by the T cell. And if the tumor cell expresses PD-L1, it can attenuate the T cell activity. However, by using anti-PD-1 antibody, we are able to block the effect of PD-L1, enhance T cell therapy, and result in immunotherapy in these patients. As you can see in the panel on the left, there are many different potential checkpoint inhibitors, and certainly time doesn't allow us to go through them. And you also see by the table on the right that, in fact, the number of tumor types in which immune checkpoint therapy is being used that are FDA approved in the United States is really quite dramatic. This is just from 2011 to 2018. Unfortunately, checkpoint inhibitor therapy, CPI treatment, although it can be terribly effective, has associated with it multiple autoimmune complications. They can be viewed as systemic autoimmune complications, as is represented here, for example, by colitis, hepatitis, etc., or neurologic complications, which can either be nonspecific or can be manifested as specific target organs or specific targeted diseases, such as inflammatory myopathies, myasthenia gravis, etc. They also can definitely trigger pre-existing autoimmune neurologic diseases, so that if a patient has had a pre-existing neurologic disease, CPI treatment can indeed exacerbate that. Let's go to the second section of the presentation, which is a look at autoimmune-related adverse events with the immunotherapy with CPI inhibition by monoclonal antibodies. And the same is true for adverse events that have been reported with small molecule inhibitors of signal transduction kinases, such as BRAF and MEK. What is the incidence of intraocular inflammation? 80 to 90% of CTLA-4 checkpoint blockade and up to 70% of patients on PD-1 or PD-L1 therapy experience ocular adverse events, most of which are mild, transient, and self-limited, but some can be severe and affect almost any organ. The incidence of intraocular inflammation as an adverse event is only about 1%, although experience is still limited at this time. Since autoimmune diseases as an adverse event of tumor therapy is caused by a general enhancement of T-cell responses, they may be an indicator of a strong and efficient anti-tumor response. We published a series of patients who developed uveitis following treatment with CTLA-4 and PD-1 checkpoint inhibition. The majority, and this was published in Ocular Immunology and Inflammation in 2020, the majority of patients in our survey are Caucasian, perhaps limiting applicability in other ethnicities. However, the Caucasian predominance is likely a reflection of the large number of melanoma patients, as skin neoplasms are disproportionately high in this population. Nevertheless, ocular inflammation has been reported in CPI treatment for cancer regardless of the organ of origin. All of our cases were bilateral, and most involved non-granulomatous anterior uveitis. However, subsequent reports have demonstrated that any type of ocular inflammation may be an adverse event. Routine screening of patients on checkpoint inhibitor therapy should be undertaken. However, all patients in our series were symptomatic on presentation. What is the usual clinical course in uveitis associated with CPI therapy? Checkpoint inhibitor-associated uveitis episodes were typically short in duration, a median of 20 days, with an excellent response to treatment and generally favorable clinical outcome, even in severe cases. Recurrence of uveitis occurred in only one case that had continuation of checkpoint inhibitor therapy, and only one patient did respond to corticosteroid treatment, and one patient developed glaucoma secondary to a fluocinolone acetonide Radicert implant. Is there an underlying difference in uveitis in patients with melanoma versus non-melanoma cancer? 12 of 14 patients with uveitis in this case series and 19 of 22 patients in the literature prior to 2020 had an underlying cancer diagnosis of malignant melanoma or choroidal uveal melanoma. There is a known relationship between melanoma and uveitis in the setting of VKH, and there are multiple reports of VKH-like disease in patients on CPI therapy. Is the development of VKH during CPI therapy in cutaneous melanoma prognostic? Development of VKH has been associated with strong anti-tumor response with PD-1 immunotherapy in melanoma patients and has been proposed as a clinical sign suggesting treatment efficacy similar to vitiligo. VKH-like disease may be associated with long recurrence-free intervals in patients with cutaneous malignant melanoma. Additionally, vitiligo and VKH-like uveitis have been described in other forms of cancer immunotherapy, including adoptive transfer of autologous T-reactive lymphocytes for metastatic melanoma. How do you manage uveitis in cancer patients on CPI? Development of uveitis is not a contraindication to continuing checkpoint blockade immunotherapy, as most cases of uveitis are readily treated with conventional corticosteroid therapy, but this must be evaluated on a case-by-case basis. Most patients have resolution of disease relatively quickly, with a median duration of disease of 20 days. Recurrence of uveitis was rare, and when present was associated with long-term continuation of checkpoint blockade therapy for cancer treatment. However, occasionally severe ocular damage can occur. This is an example of a 73-year-old patient who, with lung cancer, who was started on CPI therapy, and after 10 months presented with a 3-week history of bilateral ocular pain. On visual acuity, on examinations was 20/80 OU, and the patient had profound hypotony of both eyes. Panel A shows you Descemet folds in the cornea as a consequence of the profound hypotony, and this OCT of the anterior segments shows you the folds as well. In this slide, panels A and B show you anterior segment and posterior, posterior segment spectral domain OCT images. You can see that the profound hypotony was a consequence of ciliary body detachment, as well as multiple cells within the vitreous with significant vitritis. OCT images, spectral domain OCT images of the retina show, in fact, the presence of multiple choroidal folds, as well as the presence of optic disc edema that resulted as a consequence of CPI therapy. Despite very aggressive therapy in this patient that involved intravenous corticosteroids, discontinuation of CPI therapy, ultimately even virus planar vitrectomy surgery, the profound hypotony persisted and the patient's visual acuity remained at 20/80. I want to thank you for your attention, and I will be happy to answer any questions following.
Thank you. Thank you very much, Hank, for a very interesting talk. And over to you, Manfred, for controversies.
For a very short controversy. At first, thanks so much to all of you giving a great talks, but you used your time, definitely. So there's hardly any way of discussion. I think there are very few questions for Gerhild in the question and answers. If you could go to these ones, that would be very nice. Well, the controversy in general in these webinars is we should discuss in this group a few things which are not 100% described and clearly defined already in the literature. Let me ask first, the longer I was working in the field of uveitis, the less I'm using the sentence of uveitis is an autoimmune disease. Would you agree to this? Can we probably decide to call it an inflammatory disease and not to go to a very, very straightforward situation of autoimmune disease? Autoinflammatory to describe to a patient is very difficult, I think. So I would suggest to call it simply an inflammatory disease. What's your impression?
Well, I can tell you my impression is that, as was mentioned earlier, and first mentioned by Rachel, I think all forms of intraocular inflammation are probably a consequence of both autoinflammation and autoimmunity. And it's quite clear that there are certain intraocular inflammatory diseases that have a clinical phenotype that's very different than others. So, for example, acute anterior uveitis so frequently looks as if it could very well be an autoinflammation, whereas a disease like birdshot choroidopathy is much more like a autoimmune disease. So I think it's not unreasonable to use both terms and just to refer to it as intraocular inflammation, because I think in many cases there is both components in the disease manifestation.
I think Zuma also has shown very nicely there's always more or less, even in infections, a effect of the immune system and the infection. And when we check these situations from tuberculosis, we know that at some situations the immune response is even stronger than the infectious response. Rachel, please.
Majority of the times.
Yes. Rachel?
Rachel.
So, yes, I think that inflammatory means this is the manifestation, but autoimmune or inflammatory is the mechanism. So they're not mutually exclusive. I think they certainly both can be used to define the same disease.
I think it's even worse. I think Zumia, and there I've found also some other things, is that infection can start autoimmune responses.
Yep.
So it's a mixture, definitely a mixture of the things. Yes. So until that, I think not to take people too much in trouble, I strongly suggest to call it simply an inflammation of the eye, and not to go really to these more specific things like autoinflammation or autoimmunity.
I disagree, because mechanism dictates treatment. You can treat symptomatically, and of course that will bring the inflammation down. But if you want a more permanent kind of cure, you have to go to the underlying mechanisms, and then it becomes important if it's autoimmune or autoinflammatory or both.
Well, but I think you have to make a major distinction between what you're telling the patient and what you're telling individuals who are interested in understanding the disease and finding new treatments. As far as the patient is concerned, it's rare that I will end up having a discussion of autoimmunity. I mean, as far as the patient is concerned, their eyes are inflamed. And so saying that the eye is inflamed and not necessarily getting into whether it's autoinflammation or autoimmunity is not relevant in terms of patient care. So I would agree with Manfred that if we just talk about inflammation, whether it's intraocular inflammation or extraocular inflammation, is from a clinical perspective, the most reasonable approach.
Unfortunately, some of our patients come into the office and they say, hey, I have uveitis, it's an autoimmune disease. Can you help here? So at least one should discuss it at touch, I think, yeah.
If they bring it up, yes.
That's because many times the definitions say it is an autoimmune disease, you know. Many of our disease definitions have that word. So that's where patients pick it.
But we have to reconsider that strongly, I think, yes. Rachel gave such a lot of things, yeah. Second point, microbiome. Let me probably announce to all the participants also 2 points. There will be the next International Symposium on Uveitis organized by the ISG in Utrecht, end of August. You will find all information you need for this, for participation, which will be virtual and by person on our website. The other thing is, if you really get touched by uveitis, we can offer you the next uveitis course during Carnival in 2023. We will have the next information about that one in a few months on our website. So I cannot talk very much more about that one, but the data will be exactly the Carnival in Venice. Going back to Utrecht. Utrecht has a major topic. The topic is, what is the situation of the immune situation of classical infectious diseases? And then we analyze in non-infectious diseases the infectious causes, if there is any type of information of that an infection has started this immune response. So in that regard, this session fits perfectly in that setting. Very nicely. And one of the major points definitely will be the microbiome, of course. So we'll have talks about the microbiome in tuberculosis. Rachel will talk about the microbiome in uveitis. And we'll also talk about this in sarcoidosis. Actually, we still have an ISG project going on analyzing the microbiome of sarcoid patients with active uveitic sarcoid. So if you're interested in participating in that one, I think it's easier from Europe to participate in that one. Please contact the ISG or me. Rachel, any idea if you can influence the role of the microbiome when the uveitis has started already?
So some studies suggest that this is indeed the case. As I mentioned at the end of my talk, Phoebe Lynn's studies and Pei-Zhen studies were done in the model of induced uveitis, where you immunize mice after they had undergone microbiome manipulation. And the disease may be ongoing, and they continue to be on antibiotics in Phoebe's case, and in Pei-Zhen Yang's case, they were given a fecal microbiome transplant from patients, and this affects disease course, at least in the beginning. Now, we have confirmed Phoebe's data, and we have gone on to treat for a longer period of time. This is something that we're now preparing for publication. And in our hands, the effect on inhibition of uveitis by microbiome, when the microbiome does not serve as a trigger of disease, but rather as a modulator of disease, that effect in our hands is transient. In Pei-Zhen's study, they only went up to 14 days, so we're not sure if that affected the initial development of disease or whether that would have lasted the course. So I think this is the question is still open. How easy it's going to be to manipulate existing uveitis by changing the microbiome. I want to be optimistic and suggest that, as in other autoimmune disease models, it has proven possible by manipulating the microbiome to affect the course of disease, at least in a partial way. That may not be the only way to modulate. It may be part of a more complex therapy, perhaps a multimodal therapy. But from what we're seeing now, we should be exploring that possibility.
Oh, you get it, please.
A question to Rachel. In mice, at least you have a defined system, because the mice are usually kept under SPF conditions. They always have the same chow. Humans are very difficult because they eat different things and they are living in a quite dirty environment. When you do fecal transplantation in mice, will they sooner or later switch back to their former microbiome?
Those changes would not, I think, be permanent. They just as when you give them human microbiota, they tend to lose some of it and farm some of it. So they change the composition. In individual humans as well, there are many influences on microbiota. Why do we have a certain microbiota composition? Our HLA influences it, our diet influences it, our diurnal rhythm influences it. You name it, it influences your microbiome. So ultimately, we will tend to revert back to our own microbiome, and I believe that so will the mice. So that fecal microbiome transplant may not be a permanent solution, but it appears to be, at least in the cases where it was performed in humans clinically, to have a very good effect. Like, for example, fecal microbiome transplant for inflammatory bowel disease, that has met with some considerable success. So I think the world is looking to manipulation of microbiome as a cure-all and an end-all. That probably is not going to be the case, but it is something that needs to be explored. It's a low-hanging fruit. And it's something that is not going to have far-reaching effects, like what Hank told us, where you really have adverse effects that are really serious. So I think we should be exploring it, and we should be learning more about it.
Great. Jonas, one question to your great network. Do you think the microbiome should also belong to that network?
Yeah, so yes, you can even consider that is part of the immune network that you should study. So obviously, due to time, what I did not take into consideration in the slides, is it's more of a discussion of where to stop and where to end. So if we really talk about the molecular characteristics of the body, but yes, the microbiome of the gut is important, perhaps also of the skin in some cases, maybe even the ocular microbiome might be important for cases. And one of the strategies that's at least an underexplored field is still that we are discussing this approach of treating patients when they are symptomatic. But eventually, I'm a strong believer that using these technologies and understanding certain pathways and distribution can possibly even go to future preventive measures. But for that, we really need larger population studies and understanding how a healthy person at a certain point have a deviating, well, perhaps microbiome and becomes a patient to that. So I really think also a part of the solution may be eventually preventative measures, perhaps also for the microbiome or in other ways. So is it part of it? Yeah, so I think it is, but it's really up to discussion what isn't part of the immune system. Because if you ask me, where is the immune system in your body? To me, it's everywhere.
Sure.
Including the microbiome.
Yes. So in case you want to go to some other smaller groups of uveitis subtypes, it may be useful to contact the ISG and to form some kind of groups there, because with 150 people now, we can really get a lot of even rare diseases together and send you the information about this one. Send you a serum or something like that one.
Yeah, perhaps so. That's one thing. But so I would even if we had this discussion about autoinflammatory and autoimmune, so in rheumatology there was already this definition of these intermediate diseases that have the characteristic of both, for lack of a better term. But I would say probably even studying other, let's call them for now, inflammatory conditions that have the same characteristics. Because one of the things that really strikes out, if you look at the genetics of non-infectious uveitis, it seems to actually strongly overlap with conditions that are systemic inflammatory conditions that have uveitis. So you perhaps studying also uveitis together with all sorts of different inflammatory conditions may reveal and overcome some of the questions to better treat patients in the future. That's at least, I'm a strong believer of that.
It's an amazing topic, and you will be also presenting at the ISU in Utrecht, which is your hometown, I think. And finally, Hank.
Well, I was going to make a comment, but if you have a question, I'll be happy to respond to that. But I'll just make a brief comment.
Sure.
It's fascinating how there is nothing new under the sun, because we are talking about microbiome now. In 1950s and 1960s, almost all patients who had acute anterior uveitis were thought to have it as a consequence of an infection in their gut. And so their stool was being cultured, and some correlation was found occasionally with an infectious pathogen. And so there were a number of uveitis experts who were, in fact, offering a change in diet and other therapies to be able to cure acute anterior uveitis. And they may have been focusing on the wrong component of innate immunity, but clearly innate immunity has been recognized in some respect for many, many, many decades.
Well, I think nothing has changed since a long time when people thought this is an infectious disease. And I think a lot of our uveitis is probably primarily or secondary induced by some kind of infections of something which goes on at different points, not in the eye. Yes. But may I?
And if you go to Indian ancient system, everything is related to gut, all the diseases, and it's all about diet modification. You know, centuries ago.
If diet is the only thing, so then we have to get some consultation about diet. Difficult, I think. But
can I ask quickly, Hank, a question, which is, Hank, how about the use of checkpoint inhibitors for treating uveitis? And do you have any experience with children?
Well, I don't have experience in children with checkpoint inhibition, but in point of fact, checkpoint inhibition would presumably enhance the exacerbations of uveitis, and in fact, that's what's been observed. If a patient has a prior history of uveitis, regardless of whether it's anterior, intermediate, or posterior or pan, they have a very strong likelihood of having an exacerbation of the underlying uveitis. So I don't think that checkpoint inhibition will be helpful in that regard. Rather, what we really need to focus upon is preventing, the recognition of whatever the inciting, whatever the inciting element is that's causing the enhanced immune response, whether it be primarily innate or whether it be adaptive, that's where most of our success will probably be recognized.
So maybe we just need to broaden the definition of checkpoint inhibitors. The current checkpoint inhibitors tend to repress the immune system. We just need checkpoint inhibitors that are talking to the other side, that are going to suppress the immune system. So they're going to target the effector T cells instead of make them more fight worthy.
And yes, and that would undoubtedly result in complications such as infection and tumor, cancer, and other complications that you would encounter
always in the balance,
forming by manipulating the immune system. I mean,
there's a price for success.
Well, no, the network that was described by Jonas is a very dynamic system. And as we all know, the immune response is very dynamic. And whether you enhance one component or inhibit a component, you undoubtedly are going to see side effects that many times may be adverse.
Gerhild?
I think there was a CTLA4 Ig, which would do exactly the opposite of the anti-CTLA4 antibody, which is used as a checkpoint inhibitor for tumors. But I think it had such severe side effects that it was not used anymore.
Not surprisingly.
Can I probably come back to Hank to your presentation? You mentioned there are some other drugs besides the checkpoint inhibitors which have similar side effects. And I think we published recently about anti-TNF alpha treatment, which also have such side effects which you can group together, and they produce sarcoid, quite clearly sarcoid, most of them actually as a first sign of sarcoid. So that's also an overreaction of the immune system. But when I wrote that article, and that's what I want to mention here, I read for the first time that having such side effects seems to be a positive effect of the anti-cancer drugs. And you mentioned that already, and I think this is extremely important, and that's the reason why people who see such patients have to be aware of this and not to overtreat these patients, not to stop the checkpoint inhibitors, but simply go on with, let's say, topical or systemic, a touch of systemic steroids, and then the whole thing is gone. I think this was very, very important, even if this is not actually regular business.
Sure. No, I would agree with you, and I think most patients do respond very well to corticosteroid therapy, even with the continuation of checkpoint inhibitors. But as you know very clearly, anytime you do immunomodulatory therapy, MIT of any type, results in adverse events, and the question is how frequent do they occur and how significant are they?
Sure.
And so it is always a very dynamic balance between the presentation of disease and the control of the disease.
So thanks to all of you very much for this interesting, amazing webinar again. If there are no other questions, we are 19 and actually 20 minutes behind a little bit. It would be very lovely if you could go on to the question and answers and try to answer some of these things as they belong, as you feel they belong to you. Otherwise, I would like to thank you so much for your very, very informative presentations, and we'll meet next time, I think, in Utrecht.
14th of May for the next webinar on Behçet's disease.
Of course, in the internet, then in May.
And then
Thanks very much for participation.
Thank you.
You're welcome. Thank you all.
Thanks.
Thank you very much.
Bye.
Bye-bye.
Bye-bye.